How to run and interpret a differential expression analysis report on Basepair. So in order to run an analysis, you'll go up to the top, click on Analyses, and click on New Analysis. Then you'll choose the differential expression pipeline. Basepair has two pipelines, one to compare between two groups and one for 3 or more group comparison. So if I want to compare two groups, I select this pipeline and I get these options. You are welcome to rename the treatment and control groups to whatever you'd like. Then you'll select your samples. You'll want to make sure that your samples have already been uploaded to Basepair. Then I'll also select my control samples. If needed, you can change any default options over here. And you'll select Run Analysis. So you'll get two reports when you run a differential expression analysis. You'll get an expression count star report and a differential expression report. In the expression count star report, you'll see the quality control steps and the trimming steps, as well as the read alignment step and expression quantification. So let's take a look at what all of these figures in the report mean. As the first step, low quality reads and adapter sequences need to be removed from the data in a step called trimming. On Basepair, the impact of trimming on read quality is commonly shown in a figure like this, and you can toggle before and after trimming. Trimming is important because it removes the poor quality parts of the read, or even the entire read if necessary, which increases the amount of useful data for downstream analyses. A higher Q value means you have better data. After trimming, reads are typically aligned to either a reference genome or transcriptome. Alignment is the algorithm to figure out which gene a read came from, and Basepair provides the popular STAR tool for alignment. Another option to note, and this requires a separate pipeline, is if your species has no reference, is to assemble your own transcriptome using Trinity, which is also offered on Basepair. For alignment, there are several metrics to pay attention to. Ideally, reads align uniquely to one place in the genome, and you generally want to see at least 70% of uniquely aligned reads. In this case, we have 90%. However, some reads may align to multiple locations, so it's unclear which gene they came from, and these are generally removed. And Basepair visualizes the read processing steps from the total starting reads to how many were trimmed to how many aligned. The next step is to quantify gene expression. The number of reads that align to each gene is quantified using a program called feature counts. However, raw read counts are not appropriate to compare expression between samples and genes. Raw read counts don't account for biases such as gene length and total number of reads in each sample. So the measure FPKM, or fragments per kilobase of exon model per million mapped reads, is a common within sample normalization method. So Basepair RNA-seq analysis pipelines provide expression in raw counts, FPKM, as well as another common normalization method called TPM. And you can find all of the data here. Next, if we take a look at the differential expression report on Basepair, so this analysis statistically tests for gene expression changes between two groups of samples. For example, this can help you find genes that change between treatment and control conditions, and Basepair uses the highly cited DSeq2 tool for performing differential expression. And this tool takes as input the raw read counts per gene and outputs log2 fold changes, p-values, and adjusted p-values, among other statistics. So the log2 fold change will tell you the magnitude of gene expression change, while the p-value and the adjusted p-value assess the statistical significance. And one thing to note is compared to the p-value, the p-adjusted value is the better metric to look at since it is less likely to produce false positives. And Basepair lets you interactively explore your differential expression results. And if you need a more complex analysis setup, Basepair can still help because in addition to comparing the two groups, as I previously mentioned before, we can accommodate more complex experimental setups like comparing 3 or more groups, time course, and nested conditions. And overall, this is a review of the differential expression analysis report on Basepair. You can continue to analyze your data with further downstream analyses. We offer PCA and GSEA analysis here. And if needed, you can go into the Info tab and download additional files for further downstream analysis.